touch on the biosamples that you guys are addressing and how complex, how microscopic these types of samples are. Some of it can be how you extract those samples, some of it can be how you transport those. But you touch on multiplexing. I'm doing this for the readers' benefit here. How challenging is that when you talk about a solid sample compared to a liquid sample?
The key difference in the solid versus the liquid technology is that the solid, a pathologist can look at the tissue itself and identify this is a cancer cell, right, due to its histology and its morphology and so forth. In the blood, you don't know that. And within that blood, you have DNA from healthy tissue that is in the bloodstream, as well as from tumor. So you have to differentiate that. And the ability to go very, very deep within the sequencing capabilities to really differentiate the tumor DNA from normal DNA is a very, very challenging task. And you know, the nice thing is that the technology today allows us to actually be able to do that, both from a testing standpoint within sequencing, but most importantly for us, through the bioinformatics. This company is both a wet lab component, but the other part is it's an artificial intelligence / bioinformatic company. And the ability to take literally millions of base pair calls and being able to put those into your computer program and have that come out with meaningful information to say, I'm positive for this mutation or normal / negative for this mutation, and do that with confidence is truly a challenging yet rewarding experience here that we've helped to create for this with this technology.
In the diagnostic world, we talk about sensitivity, specificity, and reproducibility. That is a complex theme to do. How, with you all with this decentralized approach, how are you doing that consistently?
As with all medical device, you must be able to develop your technology and your tests in a way that meets the necessary and acceptable performance criteria of the assay, including specificity, sensitivity, reproducibility. So we develop our tests under what is called a design control system with very clear study designs, with very clear metrics of requirements in order to meet the necessary thresholds to produce a test in a way that different environments, meaning different laboratories around the world, can use that and get the same answer regardless of whether I'm in China or I'm in Baltimore. So we do extensive studies here in order to create that data, and then ultimately that data itself is submitted to the FDA and other healthcare authorities around the world for approvals or clearance. So it is a significant undertaking for a diagnostic business and for PGDx in order to do the necessary studies in order to meet those performance criteria that you mentioned.
There are large hospital systems that may have their own solutions to diagnostic sequencing or to treating cancer patients. However, I want to touch again on this sort of revolutionary theme about the decentralized approach and how you partner with pharma partners. What would make a pharma partner so excited to work with PGDx and your team? And it's demonstrated in the January 2018 $75 million financing that's happened recently, which is a huge testament to your team. But what would make a pharma partner be excited to work with your company?
So the key reason that a pharma partner would be thrilled to work, I think, with PGDx is multiple. Number one, I think our business model itself of creating a decentralized platform for testing is really important to them. they need to identify the right patient, right, to say who would appropriately respond to their drug. So they want a consistent, to our discussion before about having a product that is specific and sensitive and reproducible. They want that so that they have a good, robust approach to get people tested, okay? And then if those are tested locally, that allows the doctor to get a fast answer and a reproducible answer that would allow them to determine should they go on their drug or not, okay? So that is key. Today, in a centralized format, it can take weeks to get the answer back to that oncologist. So that impacts patient care. And we're trying to transform that practice of medicine here out at PGDx by making this testing much faster for the oncologist. So that's one reason that they want to work with us. Another is that they see both the cost and the time to get their drugs to market it's very, very expensive, right? And you have to take enough shots on goals because a number of these drugs do not make it. And the best way to be able to get a drug now in this era of personalized medicine to market is typically through identifying the right patients and then see if the efficacy and safety is there for that group of patients to get it approved for their drug. So for an example, when Pfizer brought their ELK product to market, their ELK inhibitors, Elcori, they used an ELK gene test in order to get that test to market. They were able to do that much faster than other drugs because they could identify the specific population that would respond, and it was less expensive in terms of accruing the number of patients that they had to accrue, and time was much, much quicker, okay? So they see this as an opportunity to improve their ability to get their drug to market much, much earlier and much more reliably than you would in a traditional phase 1, phase 2, phase 3 drug program where you're taking 7 to 12 years to bring that drug to market. You can significantly reduce that time.